Hello everyone, I welcome you all in the module 2.3 of the MOOC on Bioinformatics Essential. Now, basically, in this module, we deal with the data pre-processing. But before moving with this module, I will just revise what we have covered in the module 2.1. So basically in module 2.1 we saw about standardized formats, okay, which are the standardized formats and what is the need of that standardized formats in the bioinformatics. So in accordance with the module 2.1, in module 2.2 we saw about some different types of standardized formats, that is a FASTQ and the GenBank format. Many more formats are there. We will explore each and every one as per our need in future. Now, in this module 2.3, we will see, suppose in your standardized format, if there is any error, okay, so you will require to do the data pre-processing, okay. And what is the need of data pre-processing? Okay, so basically data pre-processing will do as per our need, that we will elaborate in this module only in upcoming slides. But yes, we'll see about mainly what is the need of data pre-processing. Okay, um, so without wasting so much time, we'll start with the data pre-processing module 2.3. So what is the data pre-processing? Okay, so data pre-processing is the process of preparing raw biological data before downstream analysis. Means before starting any procedure, what you do the pre-processing. You prepare your biological raw biological data, that is, if you download any data from database, that is called as the raw biological data. Okay, depending on the which type of database you are downloading, so that is called as the raw biological data. So basically the purpose is not to change the biology. Okay, here we are not changing the biology, here we are just cleaning our data. Okay, why we have to do this? What is the purpose of this process? The purpose is detect problems, okay, then remove inappropriate data, okay, correct formatting issues if there are any formatting issues in the data frame or in the data set, improve data consistency, and prepare data for reliable analysis. So these are the purposes for the data pre-processing. So why this pre-processing is necessary? So raw biological data sets can contain missing information, ambiguous nucleotide. Now see, in the, I guess, module 2.3, in the last or second last slide, I have mentioned about that this issue. So you can just go and watch the module 2.3. Here you can see the eye icon over. So you can go on the module 2.3C. Okay, sorry, 2.2C. Okay, my mistake. Now low quality sequence regions, adapter sequences, duplicate records, incorrect identifiers, unexpected characters, or extremely short sequences. So these are problems. This may contain in any raw biological data set. So if we ignore this problem, it will cause a problem in downstream analysis. Okay, and it will lead into misleading results. So basically, the fundamental principle behind this entire thing, behind the bioinformatics or in data analytics or anything, is if you do garbage in, then you will get the garbage out. Okay. The simple principle it is, if you input the garbage, you will get the garbage as the output only. So sophisticated computational method cannot compensate for fundamentally poor input data. In easy terms, we can say that. So what includes in this common pre-processing task? Okay, now depending on the data set, pre-processing may include, this may include one or two or all the six. First is quality assessment. We have to determine whether sequence quality is acceptable or not. Then adapter removal. So we'll remove artificial sequences introduced during library preparation by any mistake. Trimming will remove low quality sequence regions. Then filtering will remove sequences that do not meet predefined criteria. Then duplicate removal will remove the duplicate, identify and handle redundant record dates, and format validation will confirm that the file follows the expected format. Now sequence validation. Now validation asks, does the sequence file contain what is expected? Now, for suppose we are working on DNA sequence or a simple FASTA file. So it should include ATGC, the words from this only four words, right? The sequence made up of only four these words: ATGC, adenine, thymine, guanine, cytosine, right? Uh, but sometimes biological dataset can also contain ambiguity codes such as NRY, WSKM. Okay, so this character may indicate formatting errors or corrupted files or incorrect data conversion or contamination from non-sequence text by any source. I'm not pointing any source of contamination or anything, but by any mean, this can be happen in large FASTA sequences. So now see, ambiguous nucleotides. Non-ambiguous nucleotide represent uncertainty. For example, now here, suppose instead of ATGC, we got N somewhere. So N means nucleotide, and nucleotide may be A, maybe T, maybe G, or maybe C. So this is not mean that nucleotide is biological meaningless. It means that the available experimental information was insufficient to determine the exact nucleotide. Now just I am giving one example, very easy example. Now you can see this image, okay, and can anyone tell me which is the image, means which flower is indicated in this image, this blurred one, okay, left hand side, okay. So yes, you can say, sir, it's a sunflower. Why? Because you are seeing the right hand side image. But yes, uh, clearly we can't say, okay, this is the sunflower or anything, because the blur, this blur effect is not revealing the exact identity. Okay. So likewise, the ambiguous base indicates that sequence information exists, but it's uncertain. Okay, something is there, something flower is there, but we are unable to predict, or we are unable to mention that which flower it is. Okay, so this kind of thing happens with ambiguous nucleotide. Now here we have to just to be curious and be aware about this point, that data cleaning versus data manipulation. Now data cleaning is removing technical problems according to predefined criteria. Okay, data cleaning means we clean the technical problems, we find that this is the problem and we just clean that data. Okay, and data manipulation is changing data in a way that produce a preferred result. Okay, now see, manipulation is different and cleaning is different. Okay, cleaning is you have to clean the data if there is any technical problem with the file format or with the sequence or with the anything, that you have to clean. Data manipulation is in that, and it is not advised. Okay, data manipulation, in the data manipulation what happens, you do changes in a data such a way that you will get the results that you want. Okay, the preferred results you will get, not a exact or not a non-biased or unbiased result. You will get the preferred result as per your concern. You will get the results, so you change the data in a such a way that you will get the results as per your preference. So that is the data manipulation, and that you don't have to do. Now examples are acceptable: removing sequence adapters. Yes, you can remove sequence adapters, you are allowed to do it. But potentially problematic is removing observations simply because they contradict a hypothesis. Okay, suppose if any observation is contradicting your hypothesis, you don't have to remove it. Okay, this introduces an important connection between data pre-processing and research ethics. So this is very important that you should be ethical while data cleaning. You don't have to data manipulate, you have to data clean, you have to data pre-process. Okay, so that's it for this module. It's a very short module just to introduce with the what are the steps in data pre-processing, what is the need of data pre-processing. Now in the next module, that is module 2.4, we'll visit the Uniprot KB and we'll do the demonstrations of Uniprot KB. And in addition to that, in the supplementary module, we'll see about the databases, more about the databases. Already we have just covered the introductory phase of databases in module 1.4, I guess, in the first week. But in this week, uh, in the supplementary modules, we'll learn about the databases, more about the databases. So till then, thank you. If you have any doubt, I have shared the Google form for doubt raising in a group or also on a mail. So you can just raise your doubt and get in touch with me. Thank you.